Now look at this.
What's up, everybody? Penguins. Today we do topic 5.4 on non-Mendelian genetics. So your cells, you have 23 different pairs of chromosomes. 22 of them are going to be autosomes, so those are just like the normal chromosomes. And then one of your chromosomes, or one pair, is going to be called allosomes. These are the ones that determine sex. If there are two genes on the same chromosome, we'll call these linked traits or linked genes, because you're going to inherit both of these traits together on that same chromosome. Due to crossing over during prophase 1, we are able to make recombinations of this, so you might end up with some recombinant phenotypes. Traditionally, we'll find most of the offspring will be the parental phenotypes, with some of the offspring being those recombinants. So this is how we can figure out how far apart two different genes are on the same chromosome. So if you look at this, we have two that are going to be parental, so look just like the two parents. So there's the heterozygous individual and the homozygous recessive. And then there's two that are going to be recombinant phenotypes. So this is some new combination. So vestigial wings with red eyes, or normal wings with white eyes. So this is kind of looking at a new combination than what we saw in our parents. To figure out how far apart they are, you're going to find the percent recombination. The number that have recombined over the total gives you percent recombination, and the percent is the same as your map units. So if you have 14%, that's 14 map units. So when we traditionally call something sex-linked, we're saying this on the X chromosome. Now, your nucleus isn't the only place where we have DNA inside the cell. If you remember, the mitochondria and the chloroplast both have DNA. Why does the mitochondria and the chloroplast have DNA? If you remember, mitochondria and the chloroplast are actually prokaryotes that live inside of our cells, and a symbiotic theory. So we can follow the inheritance of this. If we find that something is on the mitochondrial DNA or the chloroplast DNA, we can see that it actually was inherited from the egg producer, because of the fact that your mitochondria and your chloroplast are going to be found in the egg. So it seems like every single year there's some type of pedigree question. So I think it's important that we can quickly identify different characteristics of a pedigree that might help us figure out the inheritance. Autosomal dominance, kind of hard to spot, but you'll notice here that we have two affected individuals that have an unaffected offspring, which tells us the trait cannot be recessive because there's no way to hide a dominant allele. Versus with autosomal recessive, it can be hidden. So you could have an affected individuals from two unaffected parents. So X-linked dominance, kind of hard to spot also. You're going to see that we will have 100% incidence of affected daughters from an affected male. If you remember, the male only has the X chromosome to donate to its daughters. So X-linked recessive, you see that if you have affected mother, you have affected sons because they get it from their mother.